So good morning to everyone again. Please take a seat. I think that we should start. My name is Luca Malcovati from Pavia, Italy, and I'm pleased to introduce my co-chair, Véronique Gelsi-Boyer from the Institute Pauli Calmette here in Marseille. And, we are going to have an interesting session and with questions at the end of the whole session. And for those attendees who are just attending online virtually, there is also the option to send in questions by the chat, so please free to text questions and we will read to the audience. So the first speaker this morning is Irene Ganan-Gomez from the Anderson Cancer Center, who will discuss about single-cell data.
Good morning, everyone. First, I want to thank the organizers for inviting me to speak at this conference. And in the next 15 minutes, hopefully 15, I will discuss the work that we're doing at the Clonal lab using single-cell technologies to characterize the cellular and molecular processes that drive ineffective hematopoiesis at the onset of MDS. Okay, sorry, the pointer. Oh, there we go. I have nothing to disclose. And I'm going to introduce clonal hematopoiesis here for the third or fourth time. It's probably unnecessary. But as you all know, this is an age-associated process by which blood cells become enriched in cells that are derived from a single or just a few clones harboring genetic alterations. And this is generally accepted to occur or to be detectable in at least 10% individuals at the age of 70 years old. But as it has been also discussed in the previous session, this percentage can actually be much higher depending on the sensitivity of the sequencing technique that is used to detect the mutant cells. Clonal hematopoiesis is originated by the acquisition of genetic lesions in primitive hematopoietic stem cells, or HSCs, which then gain a fitness advantage and end up outcompeting normal cells in the context of diverse types of stress. And although they are otherwise healthy, individuals with clonal hematopoiesis are at higher risk of developing myeloid malignancies and also cardiovascular disease. Okay, there we go, sorry. So individuals with CHIP higher risk of developing MDS or AML as they age, but also with the size of the mutant clone, the number and the type of mutations, and importantly, if they have cytopenias, they are also at higher risk, which is why in our studies we focus on a very specific subset of patients who are diagnosed with what is called clonal cytopenia of undetermined significance, or CCUS, which has been recently recognized by international classification systems as a precursor state of MDS. CCUS are a paradigm of pre-malignancy, and therefore they are ideal to study MDS onset and also to research early therapeutic intervention strategies for these patients. So in order to study hematopoiesis in the context of aging, we first performed an integrated single-cell RNA sequencing analysis in lineage-negative CD34 hematopoietic stem and progenitor cells, HSPCs, that we isolated from 3 young healthy donors, 4 elderly healthy donors, and 3 patients with CCUS. And we annotated the resulting clusters on the basis of their expression of lineage marker genes. So as you can see, our cells clustered forming a differentiation continuum that actually agrees with previously published differentiation trajectories. And we characterize these cells and these clusters by analyzing their frequency and their transcriptomic profiles. So we first analyzed hematopoiesis in healthy or physiological aging. And the first thing we observed, as you can see on the screen, is that elderly healthy donors had significantly increased frequencies of hematopoietic stem cells and megakaryocytic erythroid progenitors. And this occurred at the expense of the myelolymphoid progenitors. We were able to validate this data by immunophenotypic flow cytometry. And therefore, we hypothesized that this is occurring because let me see if I can use a pointer. Aged HSCs follow preferentially the megakaryocytic erythroid trajectory. We then sought to understand why these HSCs are expanded, so we performed differential gene expression analysis. And we observed that aged HSCs significantly upregulated genes that were involved in TNF-alpha-driven survival signaling through NF-kappa B. And this is indeed a known survival pathway in HSCs, and this was occurring mainly through the upregulation of MCL1, which is an anti-apoptotic gene and was one of the top upregulated genes. And MCL1 is indeed known to be essential for the survival of HSCs. And also, interestingly, these cells were significantly downregulated genes involved in proliferation and oxidative phosphorylation. So altogether, this suggested that aged HSCs were becoming more quiescent and activating survival pathways, both of which are known mechanisms for the preservation of the integrity of the stem cell pool. Then to validate the idea that this megakaryocytic erythroid bias was originated at the level of the HSCs and not a result of, for example, a depletion of the myeloid progenitors, we characterize the chromatin accessibility landscape in our healthy donors using single-cell attack sequencing. And so we performed this technique in sorted immunophenotypic long-term HSCs, isolated from 2 young donors and 2 elderly healthy donors. We identified 3 different clusters inside the long-term HSCs on their basis on their transcription factor binding site activity. And the first thing we observed was that in elderly healthy donors, there was an enrichment, and let me see if I can point it on the screen, on long-term HSCs that were primed to differentiate towards the megakaryocytic erythroid lineage. So this confirmed our results, and this was happening at the expense of subset of HSCs that were more primitive. We call them non-primed because they were not primed to differentiate on the short term. But most interestingly, when we looked at this specific subset of non-primed or primitive long-term HSCs, what we saw is first that they had active binding sites for NF-kappa B, which again confirmed our single-cell RNA sequencing data, but also when we look deeper into the open chromatin regions in distal elements, we saw that those distal elements with open chromatin peaks were enriched in genes involved in platelet development. So this means that these cells that were initially non-primed, primitive HSCs, were poised for megakaryocytic differentiation in these patients. Sorry, in the healthy elderly donors. We next characterized CCUS, and when we compare them with the elderly healthy donors, we observed that we no longer saw this expansion in the HSC compartment. What we did see was significantly increased frequencies of both early and late myelolymphoid progenitors, particularly myeloid biased progenitors. And what we also were able to observe by using immunophenotypic flow cytometry was that patients with CCUS had significantly decreased total numbers of both stem cells and progenitor cells. So this myeloid bias was not occurring because the myeloid cells were expanding, but in turn, it was occurring probably because the HSCs in patients with CCUS preferentially differentiate towards the myeloid lineage at the expense of the megakaryocytic erythroid bias, lineage. And so we again looked at the stem cells to understand why this was happening. And to our surprise, we observed that the HSCs from patients with CCUS upregulated genes that were enriched in protein synthesis processes and mitochondrial metabolism. So this is a signature of cell activation. On the other hand, these cells significantly downregulated genes that were involved in NF-kappa B signaling through TNF-alpha, which, as I previously mentioned, is the signature for normal aging. So altogether, our results suggested that HSCs from patients with CCUS were escaping the normal aging phenotype, and in turn, they were becoming myeloid biased, and they were probably sustaining the high energetic demands of myelopoiesis by augmenting their translation and mitochondrial metabolism. And here I have to stop for a second to mention that one of the top upregulated genes that we saw in this analysis in the HSCs was interleukin-1 beta, which has been thoroughly discussed in the previous session. So as you can imagine, this is a very exciting finding with potential therapeutic implications, and there is indeed another instructor at the Clonal lab who is following up on this story, because interleukin-1 beta is known to induce the exit of quiescence in HSCs and also myeloid differentiation. So obviously very important in this setting. We again sought to understand whether the myeloid bias was happening at the level of the HSCs. And so we again performed single-cell attack sequencing, this time in HSPCs isolated from 4 elderly healthy donors and from patients with CCUS. We identified cluster 1 as the HSCs on the basis on their transcription factor binding site activity. And what we saw is that these HSCs had open chromatin peaks in distal elements of genes that were enriched in cytokine signaling. And particularly when we looked inside at this list of genes, as you can see here, we saw that they are enriched in myeloid development genes. So this again confirmed that HSCs from patients with CCUS were poised towards myeloid differentiation. But interestingly, the HSCs from these patients were not the only CD34 positive cells that were expressing this highly active transcriptional profile. Because when we performed differential gene expression analysis downstream of the HSCs in all the other more committed progenitor cells, we observed that there was a general upregulation of genes that were enriched in mitochondrial metabolism. And this is particularly true for cells that were falling along the myeloid differentiation trajectory as it is highlighted on the screen and the UMAP on the far right. And so we validated these results by performing mass spectrometry in CD34 positive HSPCs isolated from patients with CCUS and with elderly healthy donors. And as you can see, what we detected with this analysis is that there were significantly higher levels of metabolic derivatives of the TCA cycle in patients with CCUS. So these results nicely confirm that there is a widespread increase of metabolic oxidative metabolism throughout the major differentiation trajectory in CCUS HSPCs. Now, we questioned whether this phenotype that we were observing was exclusive to CCUS or was also maintained when patients progressed to MDS. So we performed single-cell RNA sequencing in the HSPCs from one patient who had initially a CCUS diagnosis and then progressed to low-risk MDS without a significant clonal expansion. This was important because it allowed us to confidently compare the transcriptomic profiles of the cells that had similar, the same clonal composition. And what we observed in this patient at the time of progressive disease is that, as you can see on the pie charts, cells clustered in different transcriptional clusters. There were some new clusters, which meant that there was some changes in gene expression. But when we grouped the clusters by lineage, there were really no changes in the lineage distribution of the cells. This meant there is no change in the differentiation trajectory of the cells at the time of MDS diagnosis. And we were able to validate this lack of changes in the differentiation trajectory by immunophenotypic flow cytometry in the HSPC compartment in this patient, which is the one shown on the screen, but also in 2 other patients who had CCUS and progressed to MDS. So regarding the transcriptional changes that were happening in these cells, we also observed, interestingly, that the number of differentially expressed genes in HSCs was actually pretty low, and the largest numbers of significantly differentially expressed genes were found at the level of more committed progenitor cells. We're still talking about HSPCs, but these are lineage-committed progenitors. And particularly when we look at the myelolymphoid progenitor cells, what we see is that the genes that are significantly up are again enriched in oxidative metabolism. So this means that at the time of progression to MDS, there is an exacerbation of the pre-existing phenotype at the time of CCUS. And interestingly, these cells at the time of progression, they significantly downregulated genes that were involved in DNA repair. Of course, we wanted to validate this, and unfortunately, we didn't have any more sequential samples, so we performed single-cell RNA sequencing, this time in total bone marrow mononuclear cells from 4 patients with CCUS and 3 patients, unrelated patients with low-risk MDS. And as you can see in the center of the screen, we again confirmed that the transcriptional changes were increasing with the differentiation state of the cells being significantly higher in cells from the myeloid lineage. And in these cells from the myeloid lineage, as I'm showing myeloid HSPCs here, myelomonocytic on the screen, you can see that the significantly upregulated genes again show an exacerbation of the highly metabolically active phenotype and also myeloid bias, as shown by the neutrophil degranulation pathway. So there is indeed an exacerbation of the phenotype. But most interestingly, because here we're looking at total bone marrow, so we can look at really committed cells. When we look at the myelomonocytic cells, we see that they significantly downregulate genes that are involved in interferon signaling. And when we look at erythroid cells, they downregulate genes involved in erythropoiesis. So this here indicates that at the time of MDS progression, there is not only an exacerbation of the active phenotype, but also further impaired terminal hematopoiesis. So to finalize, I hope I have conveyed in this talk that CCUS HSCs are reprogrammed to escape the phenotype of physiological aging, and they become myeloid skewed and differentiate abnormally. They may do so in part in response to the increased secretion of myelopoietic inflammatory cytokines, such as interleukin-1 beta. And to sustain the high energetic demands of myelopoiesis, these cells active oxidative metabolism and protein synthesis and processing pathways, which actually are known to further worsen the myeloid bias. So they might be contributing to the myeloid bias through the induction of stress. And most importantly, we have observed that in progression to MDS, there is an exacerbation of the pre-existing CCUS phenotype, which has important therapeutic implications because it means that patients with CCUS and early MDS stages may benefit from early therapeutic interventions that target these cellular processes. And so I'll quickly like to thank the entire Cola lab, particularly, of course, my mentor, Simona Cola, who is behind every project in the lab. On the clinical side, Dr. Garcia Manero, and I have to make a special mention to our computational analyst, Fei Yang, because his work is essential in all our projects. Thank you.
Thank you very much. So the second speaker today is Ben Ebert from Dana-Farber Cancer Center, who is discussing about CAH-associated diseases. Please, Ben.
Thank you, Luca. Appreciate the opportunity to speak here today. It's really wonderful to see our MDS community all back together at this meeting. So many happy memories from previous episodes of this meeting. So these are my disclosures, none of which are directly relevant to the presentation today. So I just wanted to start by, this has been discussed at many previous talks already today, but the different types of clonal hematopoiesis. Everything I'll talk about today is related to CHIP and CCUS, which is defined first by David Steensma, who's here, but in the recent ICC and WHO guidelines, is having a variant allele fraction of at least 2% in CCUS with a cytopenia. But there are many other types of clonal hematopoiesis that are not associated with inflammation. As was talked about by Klaus Metzler, there are tiny clones that are almost universal with age, and those are generally not associated with inflammatory phenotypes. There are MCAs, or mosaic copy number abnormalities, that are quite common. Those are generally associated with lymphoid malignancies, in particular CLL. There are lymphoid clonal hematopoiesis or point mutations that can be found, and there are the clonal hematopoiesis with unknown drivers, which can be detected in a variety of different ways, which are more difficult to detect in clinical practice. So I'll focus on the subset of clonal hematopoiesis defined as CHIP and CCUS. So as a broad view of this, as Amy DeZern mentioned, inflammatory disorders are often diagnosed prior to the diagnosis of MDS or CMML. And so we thought that if clonal hematopoiesis or CHIP or CCUS precedes the diagnosis of MDS or CMML, that inflammatory disorders may be diagnosed in the 5 years prior to that, where an enlarged clone may be driving the inflammatory cytokines and the diagnosis of a variety of inflammatory disorders. And in order to look at that at a broad scale, we could leverage the Medicare SEER database, which is SEER is the large database of cancer cases in the United States that includes MDS. And this approach has been followed previously, but is at a much larger scale now. And when we did this, we were able to find about 20,000 cases of MDS CMML. We could link all those cases to the Medicare database and get all of their diagnoses and have a matched cohort. And you can see that a number of inflammatory diseases, including cardiovascular disease, metabolic disease, musculoskeletal diseases, are associated. Many of these inflammaging phenotypes have an increase in diagnoses in the 5 years preceding diagnosis of MDS or CMML. However, there's a decreased risk of diagnosis of neurodegenerative diseases, and this complements really elegant work from Sid Jaiswal's group showing that Alzheimer's disease has a lower incidence of diagnosis in individuals with CHIP. And so there is some seemingly protective, perhaps causal effect of CHIP and CCUS on the diagnosis of neurodegenerative disease, but an increase in risk of a variety of inflammatory disorders that are associated with aging. And the specific diagnoses include heart failure, atherosclerosis, COPD, kidney disease, fatty liver disease, osteoarthritis, osteoporosis, rheumatoid arthritis, and gout. And so in each of these cases, we now have two complementary approaches showing that these are associated with these myeloid mutations. There's an association between individuals with CHIP and incident diagnosis of these disorders, and then if looking at patients with MDS or CMML, an increase in diagnosis of these disorders prior to the diagnosis of MDS and CMML. The strongest association of all the ones that I showed just now is actually chronic liver disease, and we've worked on this quite a lot recently. As you may know, chronic liver disease often starts with fatty liver disease, with hepatic steatosis that can progress to an inflammatory disorder with steatohepatitis, and that can progress to fibrosis, eventually to cirrhosis, and in some cases to hepatocellular carcinoma, and that's extremely common in the Western world in particular. So when we look at CHIP in a variety of very large databases, including the Framingham Heart Study, ERIC, and the UK Biobank, we can see a dramatic increase or dramatic association between either prevalent or incident chronic liver disease with an odds ratio in the 2 to 3 range, which is higher than we've seen for cardiovascular disease or for other phenotypes. So, and it's notable that CHIP is not associated with the development of fat in the liver, so it's not associated with fatty liver, but it is associated with chronic liver disease and the steatohepatitis phenotype more broadly. And as you can see in the graph in the upper right, this is potentially associated with quite a large percentage of cases of NASH and chronic liver disease. If you look at the individual mutations that are associated, much like we've seen in other types of inflammatory diseases, the effect size for TET2 is most dramatic. And so we see an odds ratio of about 5 for TET2 mutations in the development of chronic liver disease, which is much higher than we've seen for other inflammatory disorders. And in this case, an extremely high association with JAK2 mutations. And that may be a different pathophysiology where the increased risk of thrombosis in JAK2 mutant disease may be associated with thrombosis in the liver and an increased risk of chronic liver disease. And much like we've seen for really all of the inflammatory disorders we've looked at, these associations are predominantly with those with individuals with the largest clones, with a varying allele fraction of at least 0.1. And those are quite large clones, again, since these are heterozygous mutations. That's about 20% of all the peripheral blood cells that bear one of these mutations. And if these are restricted largely to myeloid cells, an even larger percentage of myeloid cells. So those individuals with a VAF of at least 0.1, and particularly those with TET2 or JAK2 mutations, drive much of this phenotype. We had the opportunity to look not just at diagnosis codes, ICD-9, ICD-10 codes, but also at data from imaging unbiased by whatever diagnosis that individual might have. And there were MRIs on a number of cases in the UK Biobank that we could look for an association with CHIP and CCUS. And you can see that in the left graph that there was an association between liver inflammation diagnosed by MRI and the presence of CHIP. But there's no association, once again, with the presence of CHIP in fatty liver. So the accumulation of fat in the liver is not associated by CHIP, but the likelihood of progressing to inflammation is. We could model this in mouse models. I won't go through this in detail, but there are particular diets that will accelerate liver inflammation and fibrosis in mice, and it's a diet that's choline deficient and amino acid defined fatty diet. And this over a sort of 12 to 20 week period will lead to steatohepatitis in mice. And in mice that are transplanted with TET2 knockout bone marrow cells compared to mice that are transplanted with wild type cells, you see an acceleration of the liver phenotype. You can see in just the mice with the TET2 knockout cells, you see an inflammatory infiltrate, hepatocyte ballooning, which is a marker of hepatocyte damage, and you can see increased inflammation in mice with TET2 knockout hematopoietic cells, increased hepatocyte injury, and in an aggregate score of inflammation and hepatocyte injury, a dramatic increase in the mice with TET2 knockout. So much as we've seen with some other inflammatory phenotypes in mouse models, simply changing the hematopoietic cells to TET2 knockout will accelerate this hepatic phenotype. And then in the last story, I wanted to note a recent work on identifying the subset of individuals with CHIP who have the most dramatic worse outcomes. So a number of groups have defined features of clonal hematopoiesis that are associated with worse outcomes, in particular the diagnosis of myeloid malignancies, and that includes the particular genotype. Not all genotypes are equivalent. The clonal complexity, meaning the number of mutations that are found in individuals with CHIP or CCUS, the size of the clone or VAF, and the red cell size, and in particular red cell distribution, which we saw really in our very first report of clonal hematopoiesis, and has remained robust. So for this study, we used the UK Biobank, but we've looked at a number of other cohorts as well now, but this is a particularly large cohort with about 500,000 individuals, with the downside that they are relatively young and healthy, so the outcomes are generally much better than what we see in the clinic. So first, just looking at the genotypes, the most striking feature, as has been seen universally, is that individuals with DNMT3A mutations, which is the most common genotype in CHIP and CCUS, have an extremely low risk. They have much lower risk than anybody else with CHIP or CCUS. TET2 and ASXL1, the next most common ones, are a bit higher, but splicing factor mutations have a much, much higher risk of progressing, in particular to MDS. Individuals with the types of mutations that we see in AML, like RUNX1, IDH mutations, are at high risk, and JAK2 mutations have a very high risk of progression, in particular to myeloproliferative neoplasms, but in this particular data set, as many of the JAK2 mutation cases progress to MDS as myeloproliferative neoplasms. So we used a recursive partitioning approach to identify the individuals who are at greatest risk of progressing to myeloid malignancy. And you can see that the most important feature that distinguishes high and low risk is the genotype, the presence of high risk mutations. And actually RDW, which we collect routinely in anybody who gets a complete blood count, is the second most important feature for distinguishing the higher and lower risk patients. And then there are additional features that can refine this a bit further. But you can see that this starts to divide patients really dramatically into much, much lower and much, much higher risk individuals. At the lowest risk, individuals who have less than 1% chance of developing a myeloid malignancy in the next decade, and at the highest risk, over 50% chance of developing a myeloid malignancy in the next decade. So really separating people who maybe don't need to be seen or followed up from people who really have a dramatically higher risk. Lachelle Weeks, who led this study, then put this together into a simple risk score, really modeling the work from the IWGPM, creating a relatively simple IPSS score that has proved so enormously valuable to this field, but trying to put together a relatively simple set of features, all of which are very intuitive, into a risk scoring system, and identifying high risk, intermediate risk, and low risk individuals. And you can see that high risk individuals, which is a small percent, only 1% or so of people with CHIP or CCUS, have a very high risk of progression. And this is perhaps the set of individuals that we need to follow carefully in the clinic and perhaps focus some intervention trials on. Intermediate risk have a dramatically elevated risk, still about 35-fold elevated risk of developing a myeloid malignancy. And those with low risk have a very modest increased risk of developing a myeloid malignancy, and perhaps these are the individuals who need to be reassured and don't need to be followed in our hematology clinics, which would otherwise really overwhelm our hematology clinics if we saw everybody with low risk CHIP or CCUS. And importantly, this risk score correlates not just with the risk of developing MDS, CMML, or AML, but also with the risk of overall survival. Low risk disease, again, this is a very young, healthy cohort, so the survival is about 95% at 10 years. Low-risk disease barely changes that, just slightly lower overall survival, but intermediate risk, CHIP or CCUS, goes down to 85% overall survival, and high risk down to about 50% overall survival at 10 years. So, and much of this is independent of the diagnosis of a myeloid malignancy, so further indicating that there's a variety of morbidity and mortality associated with high-risk CHIP and CCUS that is not associated with at least a diagnosed myeloid malignancy, and we're seeing this in a variety of cohorts as well, including cohorts from Luca and from the Dana-Farber of CHIP and CCUS patients and other large cohort studies. So in summary, CHIP and CCUS are extremely common, age-associated with the risk of heme malignancy and overall mortality, as well as a variety of inflammatory disorders. I didn't talk about it today, but others have noted that these are associated with the overexpression of inflammatory cytokines, many of which are NLRP3 dependent, including IL-1 beta and IL-6. The risk of progression to malignancy and overall mortality in individuals with CHIP is associated with particular genotypes, multiple genetic abnormalities, the size of clone, altered CBC parameters, including RDW, and can be predicted with this CHRS. And wanted to highlight the work of Lachelle Weeks in the CHRS and the Medicare SEER work I talked about, and Wai-Hei Wong, who led the work on liver disease. Thank you very much.
Thank you very much, Ben.
Thank you very much. The next speaker is Luca Malcovati, who will tell us about clonal cytopenia of undetermined significance and clinical perspective.
Thank you, Veronique, and good morning to everyone. First, let me thank the scientific committee for inviting me to discuss about the clinical implication of CCUS, in particular, of high-risk CCUS in the clinical setting. So, if I can advance my slide, here we are. Recently, both the WHO classification and the ICC classification consistently introduced clinical definition of clonal hematopoiesis of indeterminate potential and the clonal cytopenia of undeterminate significance, which is characterized by an unexplained cytopenia not fulfilling any diagnostic criteria for a myeloid neoplasma, implying a bone marrow examination to exclude significant dysplasia associated with a non-MDS defining genetic abnormality. And overall, this definition will be unquestionably critical to promote harmonized diagnosis as well as to inform homogeneous clinical annotation of these cases. However, relevant area of uncertainty still remain. And we have some problem with the advance of the slide. So, in particular, the relationship between CCUS, in particular high-risk CCUS and low-risk MDS, as well as on the other side of the spectrum, the relationship between the otherwise or apparently otherwise explained cytopenia and the clone, which not always can be entirely dissected. Next, please. Okay, the next one. And finally, the uncertain classification of other hematologic disorders characterized by a clonal hematopoiesis, in particular aplastic anemia with hematopoietic clones. The next, please. As well as of other non-malignant clonal disorders, including PNH and VEXAS, as we already heard this morning. So, when looking at the genetic profiles of patients with CCUS, they unquestionably show features in between clonal hematopoiesis and overt myeloid neoplasm, in particular MDS. So consistent with their definition as a pre-malignant disorder. However, these profiles clearly implies quite marked genetic heterogeneity in terms of clone size, number of somatic mutations, as well as the distribution of mutation patterns, showing a large overlap with low-risk MDS. So in order to identify homogeneous subset of patients with CCUS, we applied unsupervised clustering analysis. Next, please. That in order to... Okay. Thank you. So in order to identify the most relevant clinical variables, previous studies consistently showed that different clone variables may be useful to identify patients with a higher risk of progression to myeloid neoplasm, including variable allele frequency, the number of somatic mutation, as well as the driver mutated genes that are indeed associated with a different evolutionary trajectory. Next, please. So this unsupervised clustering analysis identified two main groups of CCUS. The first one, next, please, is mostly characterized by isolated DNMT3A mutation with clone features not dissimilar for clonal hematopoiesis and associated with a very low, if not negligible, risk of evolution into myeloid neoplasm. Next, please. Here we are. So questioning about the true relationship between the clone and the cytopenia. The second group was characterized by isolated mutations in TET2 or ASXL1, as well as by more complex mutation patterns, including splicing factor mutation or other commutation patterns, largely overlapping with low-risk MDS and associated with a significantly higher risk of evolution into an overt myeloid neoplasm. We then question why clones with the same genetic features could be classified as either as a CCUS or a low-risk MDS. In other words, which are the most relevant clonal variables that may affect the clinical expressivity of these clones, in particular in terms of significant bone marrow dysplasia, which is, according to current criteria, the major determinant of a classification as a low-risk MDS or CCUS. So, next slide, please. We mainly identified two major factors associated with the clinical expressivity of these clones. And first, the most intuitive is the clone size. As small clones characterized by an ineffective maturation and differentiation in the context of a largely effective hematopoiesis might not result in a significant cytopenia or bone marrow dysplasia. In addition, next slide, please. The second major determinant that we identify are the biological features of driver mutations, which may result in a significantly different degree of hematopoietic impairment and resulting in a different degree of bone marrow dysplasia and peripheral blood cytopenia. And in particular, we identify two subsets of driver mutations in this context. First, driver mutations that became clinically expressed in terms of significant dysplasia around variant allele frequency of 20 to 25%. That is showing a rather strong clinical expressivity and a disturbance of hematopoiesis resulting in significant dysplasia. Next, please. And these include driver mutation patterns involving SF3B1, U2AF1, TP53, as well as more complex mutation patterns involving DTA mutations. Then we identify a second subset of driver mutation characterized by low clinical expressivity, which gain or result in a significant dysplasia for variant allele frequency around 40%, suggesting that they need to reach a clonal dominance in order to result in significant dysplasia and peripheral blood cytopenia. And this included, for instance, SRSF2 mutations, as well as DTA, isolated DTA mutations or combined epigenetic regulator mutations. Next, please. So, when looking at the distribution of these different mutation patterns in the whole cohort of patients with CCUS, next slide, please. Okay, great. Thank you very much. We found that, patterns with a strong clinical expressivity were mainly found with the variant allele frequency below 20%. Consistent with an early phase of malignant clones that do not have yet reached full clinical expressivity. Conversely, mutation patterns with mild clinical expressivity were mainly found with a largely or full clonal dominance, which, however, resulted in a usually mild dysplasia, in turn resulting in an inconsistent classification either as a CCUS or a low-risk MDS. So when looking at the evolutionary trajectory of these different mutation patterns, we found that both patterns with high clinical expressivity or mild clinical expressivity, but MDS-like, were both associated with a high risk of evolution into MDS, when accounting for competing causes of death, whereas isolated TET2 or A6L1 mutation were associated with a milder risk of evolution into overt MDS. So overall, these data suggest that bone marrow dysplasia is not a sufficiently robust criterion for the clinical staging of these clones and to properly discriminate between truly pre-malignant clones and early malignant clones in the setting of cytopenia. And so we look at the potential value of driver mutation signature to most robustly support a diagnosis of MDS. And we found that selected mutation patterns, including commutation patterns involving DTA or splicing factor mutations or TP53 mutation with additional subclonal mutations were all associated with a high specificity and high positive predictive value for a diagnosis of myelodysplastic syndrome, suggesting that these genetic signatures might be robust, sufficiently robust candidate to support a diagnosis of bona fide MDS in the context of an unexplained cytopenia. So looking at the other side of the spectrum, in low-risk MDS, overall, most of patients show quite a higher number of somatic mutations compared to patients in pre-malignant states, 2 or more, with more complex mutation patterns. However, a fraction of patients is apparently having clones driven by isolated genetic lesions, which are significantly enriched in MDS-defining genetic lesions, in particular SF3B1 and the deletion of 5q, that were proven to be able to support, to sustain a diagnosis of MDS irrespective of other commutation patterns, but a subfraction of patients with isolated genetic lesions apparently are driven by CH-like mutation patterns, including isolated DNMT3A mutation or TET2 or ERAS-X1 mutation. And when looking at the clone features of these cases, classified as a CCUS or low-risk MDS, we found that variant allele frequency is significantly different in the two distinct stages. Patients with low-risk MDS tend to have full clonal dominance, whereas at the CCUS stage, these clones usually are very small, suggesting that likely extraclonal variables may have affected the kinetic of expansion of this clone and may contribute to the clinical expressivity of these mutated clones, resulting in a diagnosis of MDS. So, as already Ben pointed out, a very nice study coordinated by Lachelle Weeks in Boston combine the most relevant clinical and genetic variables to identify hematopoietic clone associated with a higher risk of evolution into myeloid neoplasm, covering the whole spectrum of clonal hematopoiesis, including hematopoiesis, clonal hematopoiesis of indeterminate potential, as well as CCUS. And in the context of CCUS, together with the peripheral blood cytopenia that identify cases with a higher risk of evolution compared to those without clinical phenotype, high-risk mutations including splicing factor mutation, IDH1, 2, TP53 and RAX1 were significantly associated together with the variant allele frequency and the number of mutations with a significantly higher risk of evolution. And combining all these features, the scoring system is able to clearly identify a subset of high-risk CCUS, which is significantly higher risk of evolution into myeloid neoplasm, and to segregate these from patients with actually a very low risk of evolution. So clonal hematopoiesis can be also found in other non-malignant hematologic disorders, in particular in about 30 to 40% of patients with aplastic anemia, variably affecting the clinical course in terms of evolution into myeloid neoplasm and response to immunosuppressive treatments, and however, also rising relevant challenges for a differential diagnosis, in particular with hypoplastic MDS. And recent studies showed that a subset of high-risk mutations in the context of aplastic anemia is associated with a significantly higher risk of evolution into a myeloid neoplasm. So additional studies are warranted in order to understand whether these high-risk mutation signatures might, in the context of aplastic anemia, support a diagnosis of bona fide MDS, as do currently monosomy 7 or chromosomal 7 abnormalities according to current diagnostic criteria. When looking on the other side of the spectrum, that is hypoplastic MDS, in a study we conducted in collaboration with the King's College, we first found that just a fraction of patients classified as an hypoplastic MDS do have indeed a clonal hematopoiesis. So again, pointing toward the consistency of bone marrow dysplasia to support a diagnosis of MDS in the context of an hypoplastic bone marrow. In addition, when looking at the genetic signatures of patients with hypoplastic MDS and clonal hematopoiesis, we found that the mutation signatures may be extremely relevant to determine the evolution of these cases. And in particular, a fraction of patients show mutation patterns or cytogenetic abnormality highly predictive of MDS, whereas a fraction show mutation patterns clearly overlapping with clonal hematopoiesis. And when looking at the clinical course of these cases, these different mutation signature indeed discriminated two completely different groups. In particular, patients with CH-like genetic signature show a clinical course absolutely overlapping with those of classical aplastic anemia in terms of overall survival and risk of evolution, whereas cases with MDS-specific genetic signatures show a clinical course which is completely overlapping to that of more classical MDS. Suggesting again that mutation signatures might be probably more robust criteria even in this setting to identify true malignant clones. Finally, hematopoietic clones can be also found in the context of apparently otherwise explained cytopenias, and this might be incidental finding in the diagnostic workup of these patients. And in most cases, these clones show features that are absolutely overlapping with those of clonal hematopoiesis, so more likely and more consistent with passenger clones in the context of otherwise specified cytopenias. However, in a specific context, clonal hematopoietic clones seem to be higher prevalent than expected, and in particular in a recent study conducted on the Lifelines cohorts in the Netherlands, we found a significantly higher prevalence of hematopoietic clones in the context of anemia of chronic disease, which is sustained by an increased prevalence of clones driven by more common DNMT3A82 or ASX1, but also by less common and more myeloid-specific gene mutations. So pointing toward the potential relationships and interaction between cytopenia and the underlying hematopoietic clones. Finally, clonal cytopenia of undeterminate significance might represent interesting, or might offer interesting chance to early intervene on these hematopoietic clones to prevent the onset of symptomatic cytopenia and perhaps to prevent the progression, the clonal progression that is invariably result in an increased clonal complexity, increased clonal heterogeneity, that may be associated with significant resistance to current available treatments. And here you can see some of the first attempt to directly act on patients with clonal cytopenia of undeterminate significance. And with this, I stop here and thank you for your attention.
Thank you very much. So the next speaker now for selected abstract is Dr. Jason Che, who will talk about targeting Irene epigenetics to predict and overcome drug resistance in MDS and AML.
I guess my time is up, so I try to go as fast as I can. So first of all, I want to thank the scientific committee for selecting our work. Here's my disclosure. So why we're talking drug resistance? Because there is urgent clinical needs. As a hematopathologist, I can confidently say I have no problem to make diagnosis in majority MDS cases. But come to predict drug resistance, I'm useless. I cannot predict any case of drug resistance in my patient. Moreover, drug resistance contributes more than 90% of deaths in our cancer or leukemia. During the last 50 years, there are many in vitro methods have been developed to try to predict drug resistance, but none of those come to clinical use because it's low sensitivities. How will you overcome these problems? We target RNA epigenetics. Today, I'm focused talking about RNA cytosine methyltransferase and RNA cytosine methylation. You heard a lot about DNA methylation, DNA methyltransferase, but very few people know about RNA methylation and RNA methyltransferase. A couple years ago, we found two RNA methyltransferase, namely SUN1 and SUN2, interact with RNA polymerase and BRD4 and MYC to form this called active chromatin structure and messenger RNA. Basically, this structure determines drug resistance or drug response in the MDS, ML patient cells. So first, I'm going to focus on talking about novel drug predictor assays based on RNA cytosine methyltransferase. There's difference between SUN1 and SUN2. SUN1 predominantly located in the nucleus. SUN2 not only located in the nucleus, but also distributed in the ribosome and the mitochondria, involves multiple processes. We developed novel methods. The top one is plate-derived fluorescence releasing assays that can screen large number of cells very quickly and predict drug resistance. And the second assay is flow-based assays. It's very precise and quantitative assays. Let me show you the result, how this will work. You can see the top panel here. Those are drug-resistant leukemia cells, and bottom drug-sensitive leukemia cells. You can see this big difference between this drug-resistant in terms of SUN2 binding to RNA methylcytosine, and this is BRD4 polymerase, and BRD4. There's clear separation. And how this method works in the clinical samples work very nicely. You can see this patient with very low percentage blast, And we detect this almost all cells in those cells bind to NSUN2 associated with RNA cytosine methylation. We predict this patient will not respond to drug, which is venetoclax plus azacitidine. What happened to this patient? After 30 days, we did a bone marrow full of blasts. And we also found this nascent RNA synthesis can be a very sensitive method. We also developed novel technology to measure RNA synthesis. And you can see here, this drug-sensitive cells, and this assays can detect this drug sensitivity very fast, as matter as 5 minutes. And those drug-resistant cells basically do not show any inhibitions. And you can see here, different drug have a different signature or patterns of synthesis inhibition. I want to draw your attention here. This is azacitidine, decitabine. We always think this is the same kind of drug, same mechanism. You can see our data shows very different mechanism involved in these two drugs. And then we also show this is independent of apoptosis. And this method also can be used for genome-wide signature. We believe this can provide drug lineage disease and patient-specific signature for RNA synthesis inhibition. So I'm going to switch gear, talk about novel therapeutics, because when I presented these technologies, one clinician I highly respect come to me and says, Jason, I know your method, I love your method very much, but even your technology tell me the patient will not respond the drug, I'm still going to treat the patient with. I said, why? He said, I have no options. So we need a new drug. We need a new strategy. We need a new target. So this data is from Deregulation ATCG data. You can see this NSUN top one, NSUN1, and NSUN2 in all those cells, or all those, not cells, all those cancer subtypes, all increased NSUN1 and UN2 expression, especially like the lung cancer, and all this increased expression associated with poor survival in all these cancer subtypes, no matter which one you pick up. Our data shows actually, you can see the NSUN1 expression is very low, as no expression in normal bone marrow, but highly expressed in high-grade MDS and AML. So this means this large therapeutic window, and this means wide applications. But so far, no drugs have been developed to target either one of these molecules, these proteins. Why? Because there's no protein structure available for these two proteins. And you all know, but 2 years ago, but 1 year ago actually, this II predicts drug structure can be as high resolution as atomic resolutions. So in collaboration with Rick Stevenson at his organ laboratory, we use this his organ supercomputer, which is one of the most powerful computer in the world. And Dr. Stevenson predict 4 binding modules in the SNUN1, and then he designed the libraries to target those modules, and then we did a screen, and we characterizing those targets, and we move on to animal data at this moment. This is one piece of screen data you can see here. This is the lung cancer. This means inhibition. This means in the monocytic leukemia cell line, this means inhibition. You can see this couple distinct cluster here, this drug compounds here. Some of these compounds can work in all this cell line, but some only specific one of cell line. And we focus on, let me show the data of these two. And we can see these two compounds very effectively inhibit growth of those venetoclax-resistant leukemia cells. And Dr. Stevenson redocking this compound to see what's exactly binding to those molecules, those modules. He showed here that this most powerful drug actually binded both to the module A, fit into this binding curve very nicely. How this work? So we use this technology I just showed you a couple minutes ago, and we can detect how this drug work. And you can see here, this drug can disrupt this NSUN1 binding to RNA polymerase, also disrupt binding to MYC, but has no effect on the NSUN binding to 5-methylcytosine, have no effect binding to BRD4. So we know exactly this drug what target to. So now, as I said, we move on to animal model to test those compounds now. So the conclusion, we developed novel tools and identified small molecular compound which target NSUN1 and NSUN2 for predict and overcome drug resistance. Epigenetics, in my opinion, holds high promise for future diagnostics and therapeutics in cancer and leukemia. Most important slides, I want to thank lab people, especially Sean, and I also want to thank Rick Stevenson. Most important, I want to thank the Top Foundation for providing me this money for the research. Thank you very much for your attention.
Thank you very much. The next and last speaker is Masahiro Nagakawa from Kyoto University, and he will present distinct pathogenesis of clonal hematopoiesis revealed by single-cell multiomics sequencing.
Thank you for the great opportunity to make presentation today. So I'm talking about the single-cell multi-omics sequencing of clonal hematopoiesis today. So this is my COI. So clonal hematopoiesis, CH, is the expansion of clones with somatic mutations in blood from aged individuals. And it's highly associated with hematological malignancies and non-hematological disorders. And importantly, most of the functional analysis of CH have been performed with mouse models. So it's very challenging to elucidate the effect of CH mutations in human subjects because of their small clone sizes and the lack of specific markers. So this is our method. We collected samples from the patient who received hip joint replacement surgery, and we performed our single-cell genomics and gene expression profiling using CD34 positive progenitors from selected patients. It's a modified fluidigm C1HC system, so it's a microfluidics-based and automated. We added mutated region-specific primers with the same cell barcodes with those of DT primers. So we can detect mutations at any positions in the RNA, even if it's distant from the 3' position. And we also added the step of mild protease treatment for the efficient amplification of mutated regions using genomic DNA as a PCR template. This is a representative result of our single-cell genotyping. If we use only RNA, there are only limited cell fractions could be genotyped. But if we use the DNA as a template, the sensitivity is highly improved. And this is a comparison of estimated mutation cell fractions. So they're comparing our single-cell genotyping and the bulk DNA genotyping. So they showed good correlation. And first, we analyzed the phenotypes of their mutated cells compared to the wild-type cells in each patient. We chose 10 patients with heterozygous mutations with relatively small clone sizes. And we found that the mutated cells showed their enhanced expression of genes related to each of target, which is a cell proliferation markers. And importantly, the mutated cells show attenuated expression of the genes related to inflammatory response, apoptosis, and interferon gamma response. So these observations were further confirmed by using hallmark pathways and reactome pathway. And again, the mutated cells show enhanced cell proliferation and cell metabolism, and attenuated inflammatory response and interferon gamma response. So it's compatible with the clonal advantages of mutated cells over wild-type cells. And also, they may have a better fitness in their inflammatory conditions. Because CH is more frequently found in their aged bone marrow, so we analyzed if the aged bone marrow show more enhanced inflammatory conditions using only CH negative patients. And we found that their aged bone marrow cells showed enhanced inflammation, including IL-2, TGF-beta, IL-6, and inflammatory response. So since their mutated cells show the, attenuated inflammatory response, so their aging-associated inflammatory signature may contribute to the clonal expansion of the mutated cells. Then we analyzed if their bone marrow environment in CH-positive cases were further altered more than just their aging effect. So to analyze that, we compare the wild-type cells from the CH-positive cases and those from CH-negative cases in age-matched. And we found that the wild-type cells from TET2 mutated cases show their enhanced expression of interferon gamma response and also their E2F target cell proliferation marker. So those wild-type cells in TET2 mutated cases may be altered if compared to the age-matched CH-negative cases. Those were further confirmed using their hallmark pathways, and the wild-type cells from TET2 CH cases showed enhanced cell proliferation and cell metabolism, and also their interferon response were activated. So bone marrow environment of TET2 mutated cases may be altered. So our next question was if the TET2, the existence of TET2 mutated cells can cause this environmental change in the bone marrow. To analyze that, we performed competitive bone marrow transplantation using TET2 conditional knockout mice to mimic their human clonal hematopoiesis. we made the mouse which have the mixture of the wild-type cells and TET2 knockout cells, and compare them with the control wild-type cells and the wild-type cells mixture. And we sorted wild-type cells using LY 5.1 marker and performed single-cell sequencing. And finally, compared the phenotype of these mouse model with their human TET2 mutated cases. So this is the red one is a mouse phenotype, and the blue one is a human phenotype. Importantly, the mouse model also showed the enhanced expression of the cell proliferation and interferon response related genes. So we further analyzed if our mouse model is similar to the human TET2 mutated cases. We analyzed 1,300 of the reactome pathways, and here showed that there are significant overlapping of upregulated pathways, including their interferon signaling and cell cycle related genes. So these results imply that the wild-type phenotype we saw in the TET2 mutated cases may be caused by the existence of TET2 mutated cells. So this is my summary. So mutated cells showed enhanced cell proliferation and attenuated inflammatory response. And aged bone marrow showed the enhanced inflammatory response. And mutated cells may have non-cell autonomous effect. We found their enhanced cell proliferation and interferon response in the wild-type cells in TET2 mutated cases, and also the wild-type cells transplanted with TET2 knockout mice. So these non-cell autonomous effects, along with the aged environment, might be responsible for the positive selection of CH clones and contribute to the pathogenesis of CH. So these are my contributors. Thank you for the listening.
Thank you for your presentation.
So thank you. And I invite all the speakers to reach us on the podium for a brief session of questions and short answers. And I again invite people virtually attending to send in their questions through the chat. And please go to the microphone for questions from the audience. Actually, we have a first question from a virtual audience, and this is from Seisho Gawa to Ben Ebert. Ben. So, and is dealing with the reduced risk of DNMT3A mutations for the development of myeloid neoplasms. This may correlate with the overrepresentation of DNMT3A mutations in CH compared with the, MDS and AML. So does this suggest that DNMT3A mutations are strongly positively selected in CH development, but negatively selected for leukemogenesis?
Very interesting question, Seishi. And sorry you're not here, Seishi. So I cut out a slide. DNMT3A mutations certainly do increase the risk of developing a myeloid malignancy. The slide that I showed, the forest plot of relative risk of developing myeloid malignancy with different mutations, was within the population of individuals with CHIP or CCUS. If you look at patients in general, individuals in general, having a DNMT3A mutation certainly increases the risk of developing a myeloid malignancy. It's just compared to the other mutations, it's much lower risk.
Thank you. Any question from the audience? Please.
Maybe for Ben or for you, Luca. If you look at other tissues, skin or esophagus, there are people who develop mutations with aging. And is there evidence that there's an aging phenotype? where people who develop CH may also have other mutations outside of the hematopoietic system?
It's this incredibly interesting field, and certainly in maybe all of our tissues, lung, esophagus, skin, liver, there is an accumulation of mutations. No other organ system has anything like the data that we have in the hematopoietic system, because our ability to access hundreds of thousands of cases of exome sequencing to characterize mutations. So the studies in other tissues are tiny, orders of magnitude smaller. Clearly, there are abnormalities in DNA damage response, for example, or other cancer predisposition syndromes, and those cancer predisposition syndromes tend to have accelerated premalignant mutations as well. I don't know of a systematic study that has looked at hematopoietic and solid organ premalignant mutations with a particular inherited abnormality, but it's a great question. There are clearly hematopoietic specific syndromes that lead to premalignant mutations in the blood and not other organs, but in cases of Li-Fraumeni or other DNA damage response syndromes, I wouldn't be surprised if there were mutations in other tissues as well.
Thank you. Nothing to add. I think that the genetic predisposition to CH, and yesterday we heard the very nice project by Kelly Bolton, would be probably the most rational subset to investigate the question.
And if I can ask another question, what role, if any, do you think changes in the microbiome may be playing in progression of CH to other diseases? Is there emerging data on that?
Yeah, this is a great question, and I think that we have experimental evidence that the bone marrow examination may have bone marrow environment may affect the evolution and the expansion and the evolution of CH clones. Unfortunately, so far, we do not have a reliable measure of bone marrow abnormalities in the clinical practice and in the individual patient. From a clinical point of view, we may derive patients in which the bone marrow microenvironment and abnormalities may have significantly contributed to the expansion of the clone. For instance, individuals with largely dominant clones driven by isolated DNMT3A mutations or 32 mutations seems the most robust candidate for having extra clonal variables contributing to the expansion of the clone.
Yeah, maybe I didn't say, I must have misspoken. I meant the microbiome, the gut,
microbiome.
The gut
Sorry.
microbiome in terms of,
Ben, do you have any hints?
I mean, there's a nice Nature paper from Marlies Maisel showing that in TET2 mutant mouse or TET2 knockout mice, having a more inflammatory microbiome compared to a sterile microbiome will accelerate the expansion of the TET2 mutant clones, or is even relies on that abnormal microbiome or inflammatory microbiome for TET2 expansion. So there's mouse evidence. I'm not aware of human evidence that might be the case, but I think that links to many of the discussions we've had today about whether abnormal inflammation or inflammaging may lead to clonal expansion or MDS, and microbiome would be a core part of that.
I enjoyed the session. Thanks all of you. This question's for Ken. I don't want to be a reductionist, but do you think your work basically shows that maybe TET2 specifically is the thing we should be looking for? Do you have an idea how to actually apply what you've learned to clinical trials and see them out?
So, you're the second person to ask me that. I actually I don't know that we can reliably do that. So the work of others in the past have correlated TET with response to hypomethylating agents, whereas Steve, okay, DNA methyltransferase inhibitors. But then when you start parsing data, most of those studies were mixed MDS, CMML, or both hypomethylating agents. So I think this is the first study where we see in a clean, prospective, randomized study that we start seeing that correlation and it holds. Having said that, it's a small cohort. We're talking about 127 patients. As you all know, because you've seen the study is already published, it wasn't 100% clean, right? Some of the patients had received the other drug heading into the trial. So it's, I wouldn't bet the farm yet, because sometimes we see things that track with something, but doesn't necessarily stand, the test of a predictor. So that's why I'm meeting with Rafael and Eric afterwards to see how we can validate this in another cohort.
Okay, so thank you very much. I think that we can close the session, and thank you for your attention.